Hi everybody, we're going to start on Chapter 3 information. This is the cell in your course pack. This information can be found on pages 28 and 29. I just wanted to share with you a really great link for some cell information. It's called the cell scale, and it's at this link here. It gives you an idea of how small the components that we're dealing with really are. Just want to share some background information with you, some information that is not in your course pack. The term cell was actually coined in 1670, or approximately that time period, by a man named Robert Hooke. He worked on a primitive microscope that is shown here, something similar, and looked at pieces of sliced cork through the microscope. What he saw were very tiny chambers that were similar to the room he lived in a monastery. Those rooms were called cells, and so therefore the name stuck. The term cell describes the very small building blocks that give rise to multicellular living organisms like us. This is something similar to what a person who looks through a microscope might see. These are cell walls of cork, and of course plants have slightly different cell setups than do animals. They have a rigid cell wall that you can see outlined here. And animals don't have that rigid cell wall reinforced by things like cellulose. We need to be more flexible, so most of our cells do not have this kind of heavy-duty reinforcement. You'll notice that his microscope, like this picture, doesn't give any detail inside the cell. We don't see a nucleus or any other organelles, and that may have been the case for him as well. He may have only seen the outline of those cells. Nowadays we have stains or dyes that help us to give contrast to the images we see in cytology or histology. Cytology is the study of cells, while histology is the study of tissues. So these stains or dyes may stain the tissue pinks, reds, blues, and purples, and that contrast allows for us to see the difference between the various parts with a little more ease. Otherwise, what we're looking at may simply be just transparent colored materials. So once again, this is an image of, say, plant cell wall, similar to cork, what this man may have seen in the late 1600s, and each of these individual building blocks or rooms here is termed a cell. Just a little history here. We are dealing with eukaryotes in this course. The difference between them is the presence of a true nucleus. Prokaryotes are cells that do not have a true nucleus, like this bacterial cell in the image here. Eukaryotes, U means true, and those are our cells. We actually have a membrane around our DNA, and that provides a protection for that DNA. So in this image you can see there's a nucleoid region. All this DNA in here is just laying out basically for the taking inside this bacterial cell. That's one reason that bacteria tend to mutate rather quickly, is because they do not have a nuclear membrane protecting that DNA inside the cell. Here's a picture of eukaryotic cells. They do have a true nucleus, which is the definition of a eukaryotic cell. The nucleus is this structure here in the center. And you can see it looks like it's fairly neat because, well, it has a membrane around it called a nuclear membrane. You can't really see the details of the plasma membrane, but we know it's there. This structure around the outside of the cell, that's called the plasma membrane or the cell membrane. Now, you also see cytoplasm, which is the jelly-like fluid inside of our cells. It's a place for reactions to occur. It also holds our organelles in place. These happen to be cheek cells from inside of someone's mouth, and what you can also see are some rod-shaped bacteria in here. So this is a great picture for giving us the scale of comparison, how much larger our eukaryotic cells are than those very small prokaryotic bacterial cells. Just another view of the cheek cell here, and again, this structure inside, that's the nucleus. The membrane, which is the cell membrane or plasma membrane, provides a boundary around the cell on the outside. And of course, this fluid inside here, it's a semi-fluid medium, it's called cytoplasm. Now we're going to go into detail over all three of these structures, the nucleus, the membrane, and the cytoplasm, in just a few moments. All right, so now you can join me in your course pack on page 28. Let's talk about this plasma membrane, sometimes known as the cell membrane. So this is plasma membrane structure. First of all, it's a lipid component that's basically two layers of phospholipids. If you have your worksheet handy, there's a blank for you to fill in. Phospholipids create the bilayer membrane here. So these phospholipids are oriented in such a way that their hydrophilic heads are oriented outward and their hydrophobic tails are oriented inward. So if we pick on one of these little phospholipids right there, you can see the ball-shaped head is oriented toward the outside of the cell, while the tails are oriented toward the inside of this bilayer. Right here up against it is yet another phospholipid, again, tails oriented inward and heads oriented outward. So the heads are a polar region, which means that they like water, and these tails on the inside create a non-polar region. That means that they do not like water. They are water-fearing. So that particular setup drives how items can travel through our plasma membrane. Water-based items are actually kept away because it's difficult for them to make their way through this phospholipid bilayer, considering that the inside here is actually hydrophobic. There's also a number of proteins that we see here. We see integral proteins. These are the ones that pass all the way through the entirety of the plasma membrane structure. We see peripheral proteins here as well. They are only on one side or the other. So for instance, you can see several of these proteins here. There's a peripheral one there, another one here. We could say that some of these sticking up on the outside, in fact, are peripheral proteins. All right, so some of these proteins serve different structures or functions. Some are called glycoproteins. That means that they are sticky with sugars attached to them. We also see receptor proteins, which help identify these cells as who they are. Some of these integral proteins serve as carriers or channel proteins. That means they help move substances across the plasma membrane, usually from the outside to the inside, but of course it could work both ways. Now, let me give you an analogy. The plasma membrane is similar to a sandwich. If you've got two pieces of bread and meat inside, the phospholipid heads are the sandwich bread. The meat on the inside are the nonpolar tails. We also have pieces of cholesterol stuck in there. Okay, so that could be some of the fat that's on that meat on the inside. And now, if you were to take a toothpick with an olive stuck on it and to jam it through this bread, you would run that toothpick all the way through like one of these integral proteins, and the olive stuck to your toothpick would serve as a peripheral protein. The olive doesn't travel all the way through your sandwich, right? It only sits on the outside surface of your sandwich. So this plasma membrane structure is fluid. These parts can move about where they are needed. So I don't want you to think of this as a brick wall with these items firmly placed in place. They actually can be moved where they need to go. So integral proteins may be shifted about. Glycoproteins, glycolipids can move. This is a very fluid structure. So I want you to think of this more as a swimming pool full of balloons rather than a brick wall with these static structures that cannot be moved. Also some carbohydrate structures here. We see carb chains. The glycoproteins also have some sugar attached to them. And because this outer structure here is rather sticky with sugars, we call the entire outer structure here into the extracellular fluid our glycocalyx. Of course, that prefix glyco implies sugars or something that's sweet. So a lot of the features out here on the outside help cells stick to one another. They help our body recognize these cells as our own. And there's a lot going on that help link these cells to other cells to create a network of tissues. So that's kind of where we're heading with this conversation in coming chapters. All right, so let's take a look at a phospholipid more closely. Once again, you can see the polar region is the head, and these nonpolar tails down here, they form the inside of that plasma membrane structure. So of course they're nonpolar, and that means that they are hydro what? They don't like water, they're hydrophobic. On your worksheet, let's take a look at this picture here. you may want to start off with the phospholipid bilayer, and of course letter B here is a phospholipid head. And letter C, those are the phospholipid tails. More than that, letter B represents the polar region of your phospholipids, and letter C represents the nonpolar regions of your phospholipids. Let's talk about letter D. What type of protein is this? All right, so that's an integral protein that travels all the way through the bilayer. Notice letter E only sits on one side, so that's not an integral protein. That one's peripheral. Let's see, we've also got structures sticking off on letter A. This we can collectively say forms the glycocalyx. That is a term that is commonly missed on test. It does refer to the entire outer structure of this cell, and these sticky carbohydrate-rich structures help to anchor cells, identify cells as who they are, and of course, there's more to this outer surface than we have time to discuss in this course. All right, so on your worksheet, let's talk about the function of the plasma membrane. This plasma membrane serves to provide a boundary around the cell, a place for chemical reactions to occur. We also regulate what can come and go from the cell using this plasma membrane. Like we've talked, some of these external features are receptors that allow the cell to interact with its environment, and it provides a structural support. Organelles are often anchored to this plasma membrane on the cell interior. So just wanted to draw your attention here to some of these integral proteins. And first off, how do you know they're integral? Well, they travel all the way through the plasma membrane structure. They're not sitting on simply one side or another. Some of the ones we'll discuss in this class have to do with these kinds of channel proteins like this one here. You can see it's basically an open doorway for items to travel from one side of the membrane to the other side. We also talk about this here, the role of the enzyme and how these cells help to change or modify the activities inside the cell. And we'll also talk about the cell membrane as an attachment point for the cytoskeleton. You can see that strands of protein are anchored here to that cell membrane. All right, so a lot of different functions for these integral proteins, the ones that travel all the way through the plasma membrane structure. So let's turn our attention now to the semi-fluid medium found inside your cell. It's called cytoplasm. I would think of it to be somewhat similar to the structure of Jell-O. And the cytoplasm is partly fluid, which is what we specifically call cytosol or intracellular fluid. The cytoplasm also contains organelles and solutes, and this cytoplasm serves as a place for chemical reactions to occur. It also holds these organelles inside your cell in place. So when I think of cytoplasm, I think of the consistency of what Jell-O feels like, not completely a liquid and not fully a solid either. So if you break the word down, the word cyto means cell, and plasm means shape. So that kind of gives you a clue there that this particular substance gives some shape to that cell. But it's so much more than that. In this image, you can see the entire cell. You can see the plasma membrane forming a boundary around the outside of the cell. You can also see this cytoplasm inside. And you can see a number of organelles in here as well. So let's just go back and review. What's the difference between cytosol and cytoplasm? Well, cytosol is the liquid portion of the cytoplasm. It is oftentimes called ICF, which stands for intracellular fluid. You may recall that accounts for 2/3 of our bodily fluids. If you'll flip on over to page 29 with me, let's focus on the next main structure found in almost every cell of our body, and that is the nucleus. In this image, it is found dead center. I'm going to circle it for you. Okay, we're going to zone in on that and talk more specifically about its structure and functions. So the nucleus is a combination of a couple of things. The nucleus has DNA stored on the inside. and it's surrounded by a nuclear membrane on the outside. So, DNA is really important. It's the blueprint for life. It's a guide for how to make our cells. It's a guide for how to make proteins. And you think, wow, this is a really important molecule. We should really lock it up tight and not let anything get to it. And then you look at this nuclear envelope and you see all these nuclear pores all over it, and you think, well, wait a minute, how good of a security is that nuclear membrane if you have pores all over the structure of that nuclear membrane? Well, those pores are actually there for a very important reason. The nuclear membrane or nuclear envelope actually needs to be able to allow items to leave the nucleus and travel out into the cytoplasm outside the nucleus. So imagine taking, the head of a company and locking that person up inside of a chamber and having no communication. So we've got to have the communication with the DNA inside the nucleus, and we have to run the risk of allowing these pores to let things in. But more importantly, we need those pores to let information or communications come out. So it's very critical here that these pores allow for molecules like RNA to leave the nuclear setup here. Here again, you can see this nuclear membrane or nuclear envelope. It is full of nuclear pores. On the inside of the nucleus, you can see a fluid called nucleoplasm, and you see a condensed region called the nucleolus. So if there was a party in the nucleus, that's where it would be. It's at the nucleolus. That's where a condensed region of DNA is sitting. There's proteins and DNA wrapped and wound around each other, and that's why that appears as a dark spot within the nucleus. So a nucleolus is a longer word. It's a smaller structure, though, inside the nucleus. You also see a term here called chromatin, and that is DNA in its unraveled state. It's just hanging out here in the other portions of the nucleus. So it's not in the party, right? The party here is in this nucleolus, this condensed region of DNA over here. So let's review. You have an envelope that forms the boundary around the DNA. There are pores in that nuclear envelope. There is a condensed region of DNA inside the nucleus. It's called the nucleolus, this term here. The other DNA is not in the party. It's in the chromatin, which is, just hanging out here in the nucleoplasm. And the nucleoplasm is just a semi-fluid medium inside the nucleus. So let's think about this for just a moment. If fluid is inside the nucleus, what is it called? It's called the nucleoplasm. If fluid in the cell is outside the nucleus, now what is it called? Now it's called the cytoplasm. So it depends on where the fluid is hanging out. If the fluid's inside the nucleus, the prefix is nucleoplasm, and of course outside, it's called cytoplasm. Let's talk about DNA structure. We've just finished Chapter 2 information talking about this. This should be a great review for us. We know that all nucleic acids are formed of building blocks called nucleotides. Nucleotides have three components: a sugar, a phosphate group, and a base. Well, our sugar here is called deoxyribose. Keep in mind that that O-S-E ending is a great clue. What does it tell us? O-S-E tells us we have a sugar. By the way, the D in DNA comes from deoxyribose. The phosphate group here forms the ladders of the DNA molecule. So we have sugars and we have phosphates repeating over and over that make up the backbone or the sides of the ladder of DNA. There are 4 nitrogen-containing bases. Each nucleotide has one of these 4. A stands for adenine. What does T stand for? Thymine. C, cytosine. G is guanine. So let's review. A is adenine. T is thymine. C is cytosine. And G is guanine. Okay, so each one of these nucleotides has a letter. And they form the rungs of the ladder. All right, so just to review, sugar, phosphate, and base make up a single nucleotide. Here's another sugar, phosphate, and a base. So when we build DNA, we have to include a nucleotide that has a sugar, a phosphate group, and a base. Let's talk for a moment about base pairing rules. Adenines and thymines are built to pair together, so A's and T's or T's and A's go together. Cytosine and guanine are also built to go together, so C's and G's, G's and C's go together. Never, never should we see A's go with C's or A's and G's. That causes a mutation that's problematic potentially. And we shouldn't see T's going with these other letters. Only A's and T's, T's and A's. Likewise, G's and C's, C's and G's should pair together. So it is in this alphabetical structure here that we find the genetic information stored in the double helix of DNA. So here you can see the DNA strand. The backbone here is built up of repeating sugars and phosphates. And the bases here, something like adenine will pair with thymine. Cytosine will pair with guanine. Let's put guanine there. It will pair with cytosine and so forth. This DNA strand is wrapped very tightly up into a chromosome. And you can see in this picture that chromosome may in fact be hanging out in that condensed party spot called the nucleolus inside the nucleus. All right, so the rest of that DNA inside the nucleus, it's just going to be hanging there as chromatin. Just another look of DNA here. The backbone, of course, is made up of repeating sugars and phosphate groups. Okay, and then, of course, here's an adenine there. It should be paired with a thymine. There's a cytosine there. It should be paired with a guanine and so forth. Why do you think DNA is just not existing as a regular ladder inside the nucleus? Why wind up in a spiral staircase or a double helix like we can see here? Well, it's about space. We don't have room for DNA to lay out completely flat, so it is wound into this twisted shape here we call a double helix, just like a spiral staircase. And this allows for us to store that DNA for your entire body's directions in the nucleus of a cell, something so small you cannot see it with the naked eye. Once again, you can see the backbone of your DNA molecule is made up of repeating what? Sugars and phosphates, sugars and phosphates. S is sugar, P is phosphate. What is C? That's cytosine, right? That's one of our bases. So right there is a nucleotide of DNA. What does C always pair with? It always pairs with G. There's the nucleotide over there. A sugar, a phosphate, a base. So you might imagine the rungs of the ladder here are made up of base pairs. It's always, always C's and G's, G's and C's, and likewise A's and T's, T's and A's. Those base pairs are held together by hydrogen bonds. You should recognize these dotted lines here as being hydrogen bonds. They're somewhat weak bonds, but they're strong enough to allow these base pairs to hook together.